Well, thank you very much for joining us on this spotlight interview. We are with Dr. Alan Price, who holds a PhD in physics from the University of Washington and a Bachelor in Science in Physics, from California Institute of Technology. His research has spanned from the physics of liquid crystals to structural biology, and most recently, to single molecule biophysics. He has worked at the Lawrence Berkeley National Lab, St. Jude Children's Research Hospital, and the Novartis Institute of Biomedical Research. Currently, he is Associate Professor of Physics at Emmanuel College. His current professional focus is on studying the mechanism of restriction endonucleases and also in training of young scientists. Today, we will be discussing his recent PLOS ONE Lab Protocol paper entitled A Programmable DNA Roadblock System Using dCas9 and Multivalent Target Sites and the Associated Protocol in protocols.io. Thank you, Alan, for being here with us.
Thank you for that lovely introduction.
Can you start by briefly explaining what the protocol is about, how to use it, and why it is relevant?
Sure. So our method creates a programmable DNA roadblock using dCas9. So dCas9 is a cleavage deficient mutant of the Cas9 protein that's used, in gene editing. The protocol includes the design of the target roadblock sequence, design and synthesis of the guide RNA, as well as the activation of dCas9 and binding to the DNA roadblock sites. So roadblocks in general are used to interfere with translocation of proteins along DNA. These could be, motor proteins like polymerases or proteins that just diffuse one-dimensionally along DNA. And we use our roadblocks to study DNA target search in our lab. But because so many proteins move along DNA, the method has wide applicability.
And can you tell us what was the motivation to optimize this protocol? In other words, why were previously published methods not working for your needs?
Right. So one of the issues that we had with roadblocks was really the validation of the roadblock. Validating that it's properly bound, 100% of the DNA has roadblocks. So that really motivated us to look for solutions to that. And the solution in our case was designing the roadblock site to contain other functions in it. So instead of targeting a pre-existing site in our DNA, we created a new site which we put into our DNA. And in our case, we had restriction sites hidden under the roadblock so that when the roadblock is bound, the restriction sites are occluded. This allowed us to validate, by using restriction endonucleases, we could then validate the proper binding of the roadblock.
And you work on optimizing the protocol to make it work. What was the motivation? Like, why do you go the extra mile and made it public in the platform?
Well, I thought the idea was something worth sharing, and because I know many, first of all, many groups had used roadblocks and used dCas9 as a roadblock, but I hadn't seen the particular method we had used for validation of the roadblock. In addition, going for the published lab protocol was really important for my students to participate. So I do research at a primarily undergraduate institution. So we have only undergraduates, no graduate students or postdocs. And finding the right place to publish our work can be challenging. And protocols.io, for example, is a really good way we found to get our students, work out and peer reviewed.
And then, since you're now mentioning peer review, you combine the deposition in protocols.io with the peer review in PLOS ONE. And what was the motivation to take that extra step? Because we all know, it is sort of a validation, but it can also be a hurdle and time consuming. So what was the motivation to go through the process of formal peer reviewing this protocol?
Yeah. So there are really a couple of reasons. One really has to do with the external validation. So it's really important. I mentioned that we're at an undergraduate institution. So we're not typically working with a lot of colleagues in the area. It can be more challenging for us to get feedback. So that peer review is actually extremely important to help us get external validation and to refine our methods. So it's that important part of the feedback and external validation. Those are the main motivations, really.
Was the feedback that you received from your reviewers in PLOS ONE helpful in shaping the final version of the protocol? And if so, what was the specific feedback?
Yeah, so it was actually, we were lucky to have some very good reviewers who were real helpful. And although there were no changes to our actual method, they did highlight some areas where we could include more details in the protocol, specific sequences, et cetera. And that, I think, really improves the reproducibility of the protocol. So I think they really did help to improve it.
And why did you decide making the peer review history public together with your paper?
Yeah, so that's, I really believe in that. I think that that's a really good move in the publishing industry. This openness, I believe in open access to information. So really on matter of principle, when that's an option, I always choose that because I think that that really helps other people understand the validity and the thinking in the process. I think it adds a level of, validation and openness. So I am in favor of that whenever possible.
Thank you. Yeah. So we have discussed so far, the motivations of embracing open science, but I would like us now to go deep into the details of the method itself. So can you tell me, while developing the method, whether there was anything that you tried but didn't work and didn't make it to the final version of the protocol?
Yeah, that's a really good question because that's always left out of every publication, right? What didn't work. So part of my philosophy is, to try to piece together pre-existing kits and methods in new and creative ways. So we made a lot of use of commercial kits, commercially purchased synthetic DNA, in order to, avoid a lot of the guesswork. So optimize some of the issues are optimizing the, matching up these kits to make sure that they function well together. I think that in terms of the overall technique, the one thing that we had to work a bit on was the validation method. So I mentioned that that the title of the article is has multivalent, and I was looking for a word to describe the fact that the target site has other functions. So it binds the roadblock, but also we have restriction sites programmed into it so that when the roadblock is not there, we can cleave the DNA and we can read that out. So, and that's what we use for validation. So we programmed several different sites in there and experimented with different ones that were the most active within the conditions within which we were doing our experiment. So playing around with that and finding the right enzyme was key. And that process was not really, I didn't really explain that process. Of course, we just showed the one we used, but that was key to getting to work.
For the new users of the protocol, what do you think are the most critical steps?
I think, like I said, a lot of the steps are pretty straightforward, but if you've never worked with RNA, doing the in vitro transcription is pretty critical to get correct. RNA is an unstable molecule, and you have to be very careful. Cleaning off all your pipettes, washing surfaces, maintaining very, high standards of cleanliness to avoid the contamination and degradation. That's probably the most important step.
Since the time of publication, have you had to troubleshoot the protocol, things that stopped working as well as they used to do, or have you done any further optimization?
So the way we designed it, it's pretty flexible method. You can change the target and purpose just by changing the sequence, the target sequence that you're using. But the rest of the steps remain relatively unchanged. So we've actually applied it to a couple of different projects since the original publication, and have, knock on wood, not had any serious problems. It seems to be working very well. We've tried, we've even been able to use it in some new applications that we hadn't thought of before. So, so we haven't had to modify it too much.
That's really fantastic. And now, like moving back again and looking at the whole package of protocol and paper, can you tell us what has been the significance of getting your protocol out, having it peer-reviewed and published as an indexed paper, both for you and for your collaborators in terms of your career?
Yeah, well, personally, I think it gets back to, external validation. That's very important particularly for me at an undergraduate institution. We don't have a lot of scientists doing research. There's no one here doing research in my area. And my colleagues, our administration, wants to see that external validation, so that getting this lab protocol published, I think, is a really important step. And I think it actually has more significance maybe at a small institution like my own than it might have at a larger institution.
And was it exciting for your collaborators to participate in this enterprise?
Yeah, I think, for this one, this was done completely, most of the work, hands-on work, was done by the undergraduate students working in the laboratory. And they got a lot out of this project and writing the protocol. I think that, getting involved in scientific writing is very difficult. I mean, scientific writing is a difficult skill. Undergraduates who are introduced to it typically have a lot of trouble. But preparing a protocol, which is something that they have a lot of practice with, getting that to the level that it can be published and being involved in this process was a really great experience for them. They really grew professionally, I think, in this also.
So thank you so much, Alan. And before we wrap up, is there anything else that you would like to mention or discuss that we haven't touched during this conversation?
No, I think we've covered everything. I just want to say I really enjoyed the process. I think it was really helpful, and I encourage other people to submit.
Thank you so much.